Hello, everyone, and welcome to this 15th episode of Techcyte's Pioneers in Digital Diagnostics. Today's episode is entitled From Pixels to Pathology Insights, Integrating Aiosyn's AI into the Techcyte Fusion platform. We're very happy to be with you today, and we appreciate the time you've taken to be with us. Today, I'm joined by David Tellez, who is co-founder and chief technology officer at Aiosyn. He has a background in computer science and electrical engineering and has a passion for challenges and disruptive technology. He has previous experience building scalable technology for startups as a research engineer. In addition, he holds a PhD in computational pathology with a focus on developing deep learning-based algorithms. At Aiosyn, he's responsible for the technology that powers their AI solutions, coordinating efforts between the software engineering and the AI science teams. We're happy to have him. And I'm also joined by Dr. Chen, who is the Chief Medical Officer at Techcyte. Dr. Chen is a leader in digital and computational pathology. She holds a medical degree from the Mayo Clinic Alex School of Medicine and a master's in global medicine from USC. She completed her anatomic pathology residency at Brigham and Women's Hospital and an NCI postdoctoral fellowship in computational pathology at Harvard Medical School and Mass General Brigham. Before joining Techcyte, Dr. Chen led digital pathology initiatives at a cancer pharmaceutical company, driving advancements in AI-powered diagnostics. We're very grateful to have her as well. On today's webinar, we're going to cover a brief introduction to Techcyte and the Fusion platform. We'll then move on to an introduction of Aiosyn, which will include going over their automated quality control as well as Aiosyn Mitosis, mitotic figure counting. And then Tiffany is going to give us a live demo of the integration of Aiosyn within the Fusion platform. And at the end, you can post questions throughout the whole webinar, and we will have a Q&A session to wrap it up. Let's begin with an overview of Techcyte. We're a digital diagnostics company operating in more than 40 countries, supporting over 7,000 customers. Across human, veterinary, and environmental diagnostics, our team brings deep expertise and proven impact. Our focus today is more on our human diagnostics platform, which is a scalable AI-powered system purpose-built for clinical anatomic pathology. With over 5 million AI-supported diagnoses to date and a wide range of scanner integrations, we're helping human pathology labs transform how they work efficiently, accurately, and at scale. At the heart of our offering is Techcyte Fusion. It's the first AI-powered platform that truly unifies clinical and anatomic pathology. This is key, because historically, these domains have operated in silos using separate systems and workflows. Fusion brings them together under one integrated platform. This is especially important for labs looking to streamline operations, reduce training complexity, and standardize quality. To understand why this matters, let's look at the evolution of digital pathology. In version 1.0, we digitized slides and viewed them online, but workflows remained fragmented. We still relied on disconnected LAS systems and saw limited AI use. In contrast, Digital Pathology 2.0, that's what we've built with Fusion, delivers end-to-end integration. From case management to ordering and consultation, Fusion ties in LIS and EHR, layers in predictive AI, and creates a unified open platform. The result is better outcome and better ROI. Here's what that looks like in action. Fusion isn't just a viewer or a point solution. It's an end-to-end digital experience. From image acquisition and optimization to AI algorithms, LIS integration, compliance, and even reporting, everything lives in one environment. For clinical pathologists, this means efficient AI-enhanced workflows for parasitology, bacteriology, hematology, and more. For anatomic pathologists, it means collaboration tools, consultation features, and AI support for solid tumor workups and cytology. For our CP audience, here's a closer look at the Fusion clinical pathology side. It supports everything from AFB and grand stains to heme-fish, and what makes it more powerful isn't just the AI, it's how the workflow is structured. We streamline everything from scanning to case review to LIS integration. And the result is faster turnaround times, consistency, and better use of technologist time. For AP, we've built features that support not just diagnosis, but the entire pathology lifecycle. Fusion supports frozen sections, tumor boards, education, research, and even pharma use cases. It's used by pathologists, cytotechs, histotechs, and even administrators, all in one interface. Collaboration is built in. You can annotate slides, message in-app, and access grossing, performance metrics, and AI overlays, all from a browser. It's the most connected AP experience available today. Let's look at the feature stack. Fusion supports ultra-fast image viewing for slides, EM, and grossing. It integrates with all major scanners. It supports bidirectional LIS integration. It brings in both Techcyte, third-party, and homebrew AI. The platform's browser-based, multi-monitor ready, and flexible in how you store and manage documents. These capabilities make it ideal for clinical labs, research institutions, and even teaching hospitals. And that brings us to the next chapter, how Fusion becomes even more powerful by integrating best-in-class third-party AI, such as Aiosyn's. This is a core value of our platform. It's open and extensible. Whether you're screening for parasites, grading breast cancer, or validating QC, Fusion gives you access to the tools you need within the same workflow. The integration with Aiosyn is a great example of that in action. So without further ado, I'd like to give the mic over to David.
Hi, thank you very much for the introduction, and thank you for the invitation to present here some of our work on our products. I'm David Tellez. I'm the Chief Technology Officer here at Aiosyn, and we're very excited to collaborate with Techcyte in bringing some of our AI-powered products into the clinic, into hospitals. I'm happy to show you some of the algorithms that we've integrated into the Techcyte platform. But first, let me tell you a little bit more about who we are and how long have we been around. We founded Aiosyn about 3 years ago with the goal to commercialize some of the algorithms that we had previously developed in the research group of Professor Jeroen van der Laak here in the Netherlands. Our group was really embedded into the pathology department of the Radboud UMC, which is a leading hospital here in the Netherlands. And we had the privilege to access plenty of data, but also pathologists and professionals from healthcare that really helped us to develop some of these AIs that you will see today. We're currently focusing on developing the software that powers these solutions, certifying them as medical devices, as well as integrating them in existing platforms such as the Techcyte Fusion. We really focus on the AI side of things, let's say, so we don't have our own viewer platform. We are vendor neutral, and we try to put our products in the hands of hospitals, no matter the platform that is powering them. Since 2021, we've been developing 3 main product lines with the goal of bringing AI closer to practice and to the hospital. We do have a focus on the European market for now, but we have plans to move into the US in the very short term. So briefly, I would like to introduce the 3 product areas that where we focus our efforts. I will be talking about 2 of them today. On the left, you see the breast cancer biomarker area. Basically, we develop AI-powered solutions that aid pathologists with breast cancer diagnosis. I will talk more about this in a few minutes. Secondly, in the center, we also develop software that is able to analyze and quantify specimens from kidney transplants and chronic kidney diseases. This is mainly for research purposes, and it is out of scope for the presentation today. And on the right side, you see workflow improvements, where we focus on tools that are able to perform quality control automatically based on AI, which I will discuss in the second part of my presentation. So let's get started with the first product that we have integrated in Techcyte Fusion. It is Aiosyn Mitosis Breast, and it is our product for automatically detecting mitotic figures in breast cancer samples. In a nutshell, mitotic figures are cells that are undergoing division. and we basically focus on tumor cells that are undergoing division because finding more of them is evidence for more aggressive tumors. So basically, pathologists in routine practice, pathologists have to find and count these figures in order to determine the breast cancer grading. And this is part of the standard diagnosis procedure and is performed basically everywhere around the globe. You see some examples of how these mitotic figures look like in the green squares that are shown on the screen. And already, as you can imagine from this small example, you can see that finding them can be quite hard, as it is, and it has a high inter-observer variability even between very experienced pathologists. So at this point, I have a small quiz for you. I don't know how many pathologists are present listening to us today, but I would like you to take a minute to find out if you can detect how many mitotic figures are currently present on the screen. You should see now 4 options, whether you find 2, 3, 4, or 5 or more mitotic figures. Please just take a few seconds. I already see some responses coming in. I can tell you that pathologists don't really like these tasks. At least I haven't found anyone that is really a fan of this one. So it is a good application that makes a lot of sense to automate this application with AI. First, because it's just difficult, but secondly, because it's time consuming and computers are a little bit better at these things than humans are. Let me show you the results real quick. So as we can see here, this is more or less what we expected. Results are quite distributed. Some people clicked on 2 mitosis, 3 mitosis, 4 mitosis. With the algorithm, we found 4, but I would say that 2, 3, or 4, they're probably all correct, depending on who you ask. And this is, of course, an inherent problem of mitosis detection, that there is a high inter-observer variability. And this can have a huge impact. There are millions of people, millions of women that are diagnosed annually with breast cancer, and that's what we want to focus our AI technology on helping these patients. With this figure, I just want to quickly show you that inter-observer variability can really have an impact on patient treatment. So basically, what this graph shows is that the probability of getting a particular cancer grade does depend on the center and the pathologist where the patient is diagnosed, which is not a good thing, right? The diagnosis of a patient should be independent of who performs the procedure. But what we see is that it is such a difficult task that it really depends on the training of the pathologist, on what their peers are doing and their own protocol. And what we hope is that with the help of AI, we can make mitosis counting a much more robust and objective measure. So our product is really straightforward. It basically highlights mitotic figures in the entire image. So how it works is when there is a new image that comes from the scanner, it goes into, in this case, for instance, Techcyte. We immediately process the image and produce the results that you see here on the small screen on the slide. This year, we obtained the CE mark for Aiosyn Mitosis Breast, which is kind of equivalent to an FDA approval, but in the European market. And basically what this certificate says is that the device can be used for invasive breast carcinoma of any grade and stage, and the pathologist should interpret the results as if the device behaves as a second opinion. So when you open the case and you see these outputs, you can think of it like pathologists already check the image, and you can rely on these results for your own diagnosis. I want to tell you a little bit more about the clinical study that was performed to obtain the CE mark. Radboud UMC, which is a hospital here in the Netherlands, conducted an independent multi-center study to validate the algorithm, where they asked 28 pathologists to perform mitosis detection on images that have the mitotic figure, the AI overlay, and images that do not have the overlay, and then compare the results. And basically what we observed is that when the AI was enabled, close to 30%. There was close to 30% more consistency between the gradient that pathologists provided. So that means that they tend to agree more with each other when the AI was enabled, and the same happened also with the panel of experts that graded the cases. This is of course good news. We also saw that the pathologists tend to look at the same hotspot when the AI is enabled, which also reduces the inter-observer variability that I showed you before, which is again good news for the patients. Furthermore, we also observed an increase in productivity. So pathologists were able to review more cases in the same time with the AI was enabled, which of course helps with the return on investment by the labs that wants to implement AI. And finally, we also conducted a survey at the end of the study where we asked pathologists what was their general experience with the device using AI. It was new for some of them. And basically, we got overwhelmingly positive response, which indicates that they really want to use it in practice. The good news is that the algorithm is now available at Techcyte Fusion, and we encourage users that would like to try to contact us or contact Techcyte for more information and to try it out. So that's about Aiosyn Mitosis Breast. I would like to briefly also talk about a second product that we have integrated into Techcyte Fusion, and this is Aiosyn QC. This is our tool for automated detection of image artifacts for quality control. And this tool is intended for technicians and lab managers, and it is not a medical device. And basically, what the product does is that it is able to detect images that are that have quality problems, for example, that are out of focus, or have large air bubbles or pen markers, or they're incompletely scanned, etc. And why is quality control important? Why do we care about detecting images that are problematic and that don't have enough quality for diagnosis? So first of all, pathologists need high quality images to perform an accurate diagnosis. And not only that, but if they receive images that are not good enough, there could be also delays for the patients. For instance, imagine that the pathologist received a slide that is out of focus and they're not able to perform the diagnosis in the tumor. They might have to wait days until a new image is created for them. Of course, this also applies to research and clinical trials where the results can be compromised if the image quality is not consistent or good enough. And finally, and it's becoming more and more important with the implementation of AI, is that we have to ensure that AI algorithms from any manufacturer perform correctly, because we've seen in research that unseen artifacts can severely degrade the performance of AI algorithms. So with Aiosyn QC, we want to find out those regions where the quality is not good enough and prevent them from going into AI algorithms down the line. How does it work in clinical practice, this quality control? So typically what happens is that technicians check every single image or a large percentage of them before they go into the pathologists. And they spend quite some time looking for problems like out of focus or missing tissue. If everything is fine with the image, they are sent to the pathologist for examination. And if there is a problem, they are placed on a different queue where the images are reviewed. And for instance, if it's out of focus, then it will need to be rescanned. As you can imagine, this can be a time-consuming process, and we would like to use AI and automation to make it a little bit easier for them. At this time, I would like to also challenge you with a small quiz. So what we've seen is that there is quite some variety between centers regarding the number of images that are checked for quality control. There are some labs that are not doing any quality control at all because it's quite expensive, for instance. And we also see some other hospitals that perform quality control on every single image. So I would like to know a bit more about you. Please select the option that applies to you. If you're checking every single image, please press A. If you don't perform any QC, use D, and anything in between. I already see some answers coming in. Let me show you the results. Yeah, so there is a bit of variety of answers. We do see some hospitals that don't perform any quality control. I don't know about these cases, but typically what we heard is that well, it's quite expensive to have technicians dedicated to this task. And I think this is, again, where AI can really help to accelerate and maybe focus those technicians on only the few cases that are flagged as problematic, for instance. And we also see some cases where every single image is checked. And I think there's also a good case for AI there to prevent this technician time to be wasted, let's say. Let's continue with the presentation. Yeah, so how does it work with Aiosyn QC? When we deploy this AI, so what we do is that Aiosyn QC will check every single slide before any humans opens the images. And if something is detected, so if there is low quality images found, then they're flagged and placed on a different list for review later. And this prevents that technicians are checking the majority of images that are usually high quality and have no problems, but focus on the ones that are problematic, which might be a few per day, depending a bit on the hospital's conditions. If these artifacts are found, they are displayed on the screen. For example, there will be a region of interest or a pointer pointing at the region that is out of focus, or missing tissue, or any of the other artifacts that you see here. And there could also be a dashboard where we displayed slide level statistics. So for instance, something saying like 30% of your image is out of focus, which can be quite useful for practice. So a bit of a case study that we have. So we've deployed Aiosyn QC in one of our customers here in the Netherlands, where they produce about 1,000 images per day, and they're all checked by Aiosyn QC automatically. There is nothing that the technician has to do. There's no clicks. The images come directly from the scanner into the algorithm, and the results are placed in the platform. In this particular case, there are 3 artifacts that they are looking for as out of focus, white balance deviation, and incomplete scanning. They have this on a daily basis, and Aiosyn QC helps the technicians to focus directly on those affected cases so they can put more time on fixing the problem and not so much on just browsing through images that are completely fine. And this is quite useful for them and saves a bit of time. Yeah, so in terms of the workflow, as I mentioned before, the images are coming from the scanner. In this case, they have Sectra, but it's exactly the same for Techcyte Fusion. These images are sent automatically to a platform. We analyze the results, and then we display the results on the platform. This, as I said, seems to save quite some time, and we are now expanding the functionalities of Aiosyn QC to solve other quality control problems that they have. In summary, Aiosyn QC allows you to save time, implement reproducible quality control, and monitor the quality of your lab operation more closely. And with this, I would like to conclude my presentation, and thank you very much for your attention. And let me know if there is any questions from the audience that I might be able to answer. Thanks.
Thank you, David, for that great presentation of Aiosyn and your AI. Tiffany, I'd like to turn the mic over to you for a live demo now of the Fusion platform and the Aiosyn integration. Tiffany?
Thanks, Troy. Thanks, David, for going through Aiosyn. Really excited to share what Aiosyn integration will look like within the Techcyte Fusion platform. So today I'll be showing a demo of the Aiosyn QC algorithm as well as the Aiosyn Mitosis detector for breast cancer. So we'll start with the QC algorithm. So as you can see here, this is the Techcyte work list. And upon clicking into the work list, you can see that there are cases and scans with visual indicators of whether or not it has completed QC review. And in this case has been flagged to be QC failed. So this will give me, as a user, insight that one of my slides have been identified by QC algorithm from Aiosyn as being failed. So in this case, if I click into the case, I look at the scan, and the scan looks very out of focus. And if I were to look at what the Aiosyn QC has identified, I could see that they have identified these regions as all being out of focus. So then going into the QC panel, I can see here that using AI, so the Aiosyn QC algorithm, it was able to select this scan as failing QC for out of focus. So this will save pathologists and scan techs a lot of time, just being able to screen through high volumes of cases and detect these types of scan and slide artifacts that can really just, speed up the workflow for our pathologists. So then going to the next example here, I'm going to show the Aiosyn Mitosis algorithm. So right here, we have a breast cancer slide, and you can see the tumor regions right here as well. So then going into our AI results panel, we can see there's been several algorithms run on this case, but I'm only going to show this slide right here. And like David had previously shown in his PowerPoint presentation, we now have the ability to bring in these annotations in from Aiosyn, and these are all the mitoses that have been detected by Aiosyn's mitosis detector algorithm. So this is a huge time savings for pathologists. They're nicely boxed for you. And now as you're doing your review, you can see the mitoses and help you generate your own count for grading your mitoses, for grading your breast cancer cases. So these are the two examples that I wanted to demo today, and we're really excited to be working together with Aiosyn to have these fully integrated within our system. And we look forward to having users have access to these algorithms, and we also look forward to our continued relationship with Aiosyn. So thank you so much. And back to you, Troy.
Thank you. I appreciate that. Okay, right now we have gathered several questions from our audience members. The first one that I'm seeing is, what makes Fusion different from other digital pathology platforms? Tiffany, maybe that's a question you can field.
Sure, yes. So Fusion is different. Firstly, we are one of the fastest cloud-based viewers. So you can sign in from anywhere as long as you have internet support. So because we're cloud-based, we can really leverage the usefulness of having different browsers, so we can remain context synced across different windows. So like Troy had mentioned, we're able to support multi-monitors, multi-windows, ensuring that every single window is context synced. In addition, we really focus on integration. So we integrate with the LIS, with the EHR, with third-party AIs that are able to have the same type of integration APIs with us. And with our integration, we are really able to pull in AI deeply into the workflow and to bring you those efficiency gains rather than just offering AI that doesn't have a way to clinically deploy. And then I would say for us, we really focus on driving together with the platform. So we want to make sure that the user doesn't have to open up so many windows and so many systems just to do their patient or their case review. So we're trying to bring all of that under one system, whether that's looking at clinical history from your EHR to even sending out like a interdepartmental consult or second opinion. You don't need to leave your system to request that from another user. All of that can be housed within our system.
Thanks. Bailey asks, what's required to turn on or activate the Aiosyn tools within Fusion? And maybe that's a question that both of you can handle.
Maybe Tiffany, you can go first.
Yeah, so how we are offering third-party AI is through similar to something like a marketplace. If you're familiar with like the App Store or something like that, when you're in your case, what you can do is you can launch an AI algorithm, and from there there's a selection. So then you would select which algorithm you want to run on your cases. So in this case, if you would like to run, the mitosis algorithm, you can select it. For something like QC, that is something that we will have to discuss ahead of time so we can get that to run before anyone ever, clicks or looks into the slide. And that will I think require just a conversation between Techcyte and Aiosyn as well as the lab.
And David, on Aiosyn's end?
Yeah, and I think Tiffany explained it very well. Maybe something to add is that we also support cloud, but also local deployments. So there are some customers that are also wondering about privacy, and they want to keep the computing side of things localized, and that is also perfectly supported and works fine with Techcyte. So I think we're flexible in that regard.
Actually, David, your question leads to a question or your response leads to a question that Gunther asked, which is Fusion cloud-based or can it be run on-premise as well? Tiffany, maybe you can field that question.
Yes, Fusion is cloud-based, but we are able to offer on-prem or hybrid storage. But outside of that, we're fully cloud-based. So we believe in being cloud-based and really freeing up pathologists from having to, be on site all the time or have to VPN in. We believe cloud-based can get you the same viewer performance and also still retain all the security and compliance that users may be concerned about.
Thanks. Elsa asks, what advantages does Techcyte's standard-based platform offer to third-party AI developers like Aiosyn?
Sure. Yes. For Techcyte, we're excited to partner with third-party AI, and we want to make it as easy as possible. So by being standards-based and having open APIs, this ensures that whoever wants to partner with us can really partner with us. And we're always listening to our clients too and our customers. If they have specific AI needs that can be solved by a third-party company, we would love to host that on our platform. And to do so, we've decided that we are going to be open and we're going to be standards-based.
Thanks. Alexandra asks, what regulatory approvals does Aiosyn Mitosis Breast have, and can I use it in clinical practice today?
Yeah, so Aiosyn Mitosis Breast can be used for diagnostics in Europe as of today. We received the certification earlier this year, so it's perfectly fine now. And for the US, we are applying to the FDA to get approval there. So for now, it's only available for research purposes, although the algorithm will work pretty much the same way. So you can always test it for research purposes, and then later down the line, once it gets approved, you can also use it for diagnostics.
Thanks. We've got another question for you, David. In a typical workflow, how long does it take for Aiosyn QC to process and flag a slide after scanning?
Yeah, it's a good question. Depends a bit on the hardware, but it can range between 30 seconds to 1 minute. So typically the user doesn't really see any delay. With this case that I just presented, we're able to process thousands of images per day. And if delay is a problem, we can always parallelize it with more hardware in the cloud. That's also a benefit that Tiffany mentioned, that we can really scale things up in the cloud. So typically there's not an issue for QC. QC is quite fast.
Thanks. Julian asks, can Fusion handle all case types like biopsies, cytology, and frozen sections and integrate with LAS?
Yes. Yes. So for Fusion, Fusion is going to be a platform that will be able to host different use cases like you mentioned here with cytology, surgical pathology, frozen section. So we're able to then consolidate all these types of cases under one platform. And we really see that as an advantage. For example, if you have a case that has gone through frozens and now you have the permanent, you're able to see those images side by side. Or if you have a case that has both a concurrent PAP and a cervical biopsy, you're able to then, view these images for the same patient, also within the same system. So we see that as a huge advantage, being a unified platform, able to support multiple types of imaging. And I also want to add that we're able to support electron microscopy images and fluorescent images as well. So we can support other more specialized workflows like the renal workflow. So you can keep all of your diagnostic images and slides all together.
Thanks. And I'm aware of everyone's time constraints today. I'll just field one question for you, David, more, and then one for you, Tiffany. David, does the QC mode check if there is any tissue that was missed during the scanning process, such as tissue fragments?
Yes, we have a feature available for incomplete scanning or detecting missing tissue. That what we'll do is that it will check the macro image, which is a small thumbnail that is taken from the slide, and compare it with the whole slide image to see if there is any missing tissue that the high resolution scanner has overlooked. And we can tell you at slide level, but also at precisely which pixels are missing.
Thanks. And Tiffany, Andre asks, how easy is it to customize Fusion to fit our current lab's workflow?
Yes, so Fusion is very, very flexible and customizable. So we can support, small scale pathology labs of, 1 to 2 pathologists to very, very large volume centers. How our work list is set up is you can move around any of these columns. You can, pre-select which columns you would like to show. So if you have, trainees or residents, you can incorporate in, those columns that support trainee workflows. Or if you don't, you can just remove them out. And we have the ability to save different views, and these views can be personal views or shared global views. And we have the ability to even filter out, doing smart filtering to see cases from specific subspecialty groups like dermatopathology, renal, GI, liver. Like we're able to separate all of those out. So we can really support all types of labs and institutions. And that's something that we think is a huge advantage, again, of going with Fusion.
Thanks, Tiffany. Thanks, David. And thanks to all of you for joining us. This has been episode 15 of the Techcyte Pioneers in Digital Diagnostics webinar series. And today's episode was From Pixels to Pathology Insights, integrating Aiosyn's AI into the Techcyte Fusion platform. Thanks for joining us, and have a great day. Bye-bye.
Thank you.